first protein of the day is the fungal luciferase, which is located at this uniport accession number. For simplicity, I already loaded it here. It's a pretty cool protein. It has catalytic activity converting 3-hydroxyhistidine into caffeonyl pyruvate, which emits light in the process. If we look at the sequence of this protein, we will immediately notice that there is a very large membrane slash secretion segment in the first 25 or so residues. It's very likely need to be cut off, resulting in something of the following. For the purpose of structural biology, it's not uncommon to be concerned with respect to effects of the tags and so on. And so we're designing two constructs here. One with the C-terminal of his tag is 8 in green, one with an N-terminal of his 8 in green, as followed by the TAV ANL-YFQG cleavage site. Now, those who are familiar with mammalian secretion signals will remember that not every secretion signal works with every protein. The primary culprits for that are the first 3 residues after the secretion signal cleavage site, which we can remedy quite conveniently by adding a GA sequence in front of our protein. And here are some references that will help you make your own choices. Now in benchling, we're going to clone these into our super secret proprietary p-beacon expression vector, which I've sort of done here, almost. And now I'm going to add a glycine-alanine pair. So a glycine, for example, would be GGC, and followed by an alanine, for example, GCC. That should result in insertion of glycine-alanine pair. Yep. Just need to make sure that this translation is extended into right to the beginning of the gene. Starts at 3093. For some reason benchling doesn't let us edit it, which is weird, but okay. I'll simply add another translation. I'll call it GA space here. Alright, save. Here we are. Now I need to repeat this process. Here's already a G. Tiny linker, and we're going to add another A. There we go. Now the reason for there being 2 distinct gene sequences is that behind the scenes I've done 2 codon optimization and reconciliation strategies. One from JCAT and one from benchling itself. They produce rather distinct types of optimization, and it would be curious, since we're doing this for fun, to see which one is a winner. Obviously all the other parameters like the location of the tag and so on may also impact secretion and expression of these proteins. So in reality this is a little bit of a silly experiment, but ultimately we just want a protein with a tag expressed in mammalian cells. So that's not a big deal. Alright, so now we have these 2. So our next protein is DCAF11. It's a critical component of an E3 ligase, recently discovered to be quite useful for degrading proteins. Its structure hasn't been determined, but it is fairly clear that it's a member of a WD-40 or beta propeller architecture, and this architecture is commonly found in a lot of proteins that participate in forming meshes or networks. This is a rigid scaffold that stabilizes other proteins binding to it. DDB1 is a classic example of that. Some transcription factors and so on. Many proteins have this architecture. So our goal is to determine what this protein structure actually is, for which purpose these long disordered tail ends are probably not very conducive. Obviously, we're dealing with the model here, and again, obviously for folks who are interested in sort of a purist application of prior knowledge, we're not going to really use this for our work. Instead, we're going to do secondary structure prediction, which I've done behind the scenes, secondary structure and multiple sequence alignment, which indicate that indeed these sequences marked in red probably don't have a lot of fold to them. If in a hurry, you can just look. There are basically almost no hydrophobic residues, which is not a guarantee, but a very strong indication that these residues are participating in an unfolded protein formation. Also, look, a whole bunch of charges, a whole bunch of GS. Here we have also very few hydrophobic residues and a lot of S's and T's. This is the last beta sheet, and this is the first alpha helix predicted by secondary structure algorithms. And then these residues in black, or in bold, are strongly conserved across distant, deliberately peak to distant orthologs. So there's conservation here, which indicates that maybe cutting after this F is advised. And so the construct that we can come up with is shown here. Obviously need an initiator with methionine, and the first residue of the protein, the last residues of the protein, and then a small G spacer in purple, followed by an uncleavable C-terminal his tag. If we go back to our magical p-bacon vector, we will clone between these two primers. It will be used to open the vector, more about that later. And this is a synthetic gene. It has been produced by JCAT and then re-optimized by Vector Builder because I did not like the high GC content. Doing so drops the code adaptation from 0.99 to 0.92, which is acceptable, but the GC content goes from 67 to 58%. Now we're going to go and, just for fun, design the same sequence in a context of a prokaryotic expression vector, so something like BET24. So to do that, we're going to use this modified BET24 tag vector. I am extremely lazy, so I don't like to retransform and reclone all the time. And this is a simple vector that incorporates both the PC7 and PTAC with two lac operators, which means that I can use this to be expressing proteins in any normal E. coli. So it will use the E. coli indigenous polymerase, or it can use T7. Now, of course, if my protein happens to be toxic, or my gene happens to be toxic, then I've just shot myself in the foot, because I cannot use a safe cloning strain like TH10B or TH5-alpha safely, as my protein will be slightly expressed from the stack promoter, even under the control of the lac operator. But I'm willing to live with that because I don't think decaf11 is all that toxic a priori. As you can see here, there's an enhancement expression enhancing peptide we call MPEP. Silky and I have come up with it a long time ago, and we've been using it very successfully for a number of years. And here's a nice 8 his tag, followed by, of course, our simple NL YFQG TFCOT. The sequence has been re-optimized in Vector Builder, although I could have used the benchling optimization as well. It doesn't really make much of a difference. With catnip connotation being 94%, and GC being about 54%, which is all right for E. coli. So thus we have, again, two vectors, two synthetic constructs. So our next and final protein is treZ from Nocardia opaca. It's a malto-oligosyl trehalose trehallohydratase, which in simplified terms means that it cuts off trehalose from ends of glucan. And Nocardia is a bacterium. Nocardia genus in general is found in soil, sawdust, and also in our gingiva, in the cavities next to our teeth. And it does have the potential of causing a fairly unpleasant infection, luckily treatable with antibiotics. So this enzyme is interesting mechanistically. This enzyme is interesting for biotechnology and also for the discovery of antibiotic substances. So looking at its sequence and then its predicted structure, one would recognize the alpha-beta barrel fold, which is classic for sugar hydrolysis, and also the little ear-like beta sheet attachments. However, a trained eye will notice that these beta sheets are a little bit truncated. There seems to be something missing. If we compare this with a couple of structures that are similar in function, very distant in sequence, we will notice that areas shaded in pink and red, these are two related structures, one from Deinococcus and one from some hyperthermophile, I forgot which one. This area is missing, so there's no sequence. That makes me a little bit nervous, because it's not the first time, wouldn't be the first time when I see annotated sequences that are incomplete. Sometimes annotation engine picks from the genome one of the methionine start points and not the correct one. So it's something that we should investigate. One of the first things that we do is we blast against PTB. Oh, look, there's a problem with the search. Right. Oh, after rerunning the search using only Nocardia as search field, we seem to have less scare. However, there's 16 residues missing, another 16, some more. This may be a problem. We may have to go back to the original sequence and understand better if this is in fact correct. So there's 16 residues missing. The other interesting fact to consider is that even though I sent in Nocardia opaca, I am not getting it back. Nothing. That is probably because the species is officially renamed, or subnomenclature is created as a Rhodococcus opacus. Interesting. This particular sample apparently had been collected in Moscow in 1987 by S.N. Gorlatov. So if we go to the full genome of this organism at NCBI and look up treZ, we get this locus tag. That seems to be correct. Let's see if we have a problem. So this is a case where an annotation has been made starting from GTG, which is a minor form of an nascent start codon that still translates into an ATG-like, meaning tRNA methionine in bacteria. But I'm still very suspicious. Let's see if we can dig up a little bit more. So what I did is I updated this from 850 to 800, so I stepped 55 back. Let's see if there's something going on. As you can see, when we're stepping back, we are still finding this EVD-GRRB. There are no stop codons. There's single cysteine. However, the question is, where does this thing begin? Let's step back some more. In the meantime, our Rhodococcus search came back, and here's one query subject, and here's everything else, which begins with subject 16, 16, 16, 16, 16. So this is very specific. There's query subject 1 from Rhodococcus opacus PD630. That's at least something. Another 16, 16, and 16, and so on and so forth. This looks like we have 16 residues missing. The issue, however, is that if we go from here, we quickly pass 16 residues and we don't get a methionine, and the sequence just runs on. So is it a possibility that the sequence is super long, or that the sequencing error has been happened, and the assembler just didn't know how to call it. So the annotation engine picked the nearest residue that could have been a methionine. This V changed one nucleotide, or pretended that GTG is also okay to start with, which in some bacteria sometimes it is, and called it a day. So at this point we have two options. Option one is just we take the first 16 residues of what appear to be very, very similar proteins. Look at these similarities, 96%, 97% positive, so on, and just transplant them and hope for the best. Or we're going to take a look at what this guy blasts against and see what happens. And this is good. Look at that. Our query produced a perfect match, with the exception of this M being a V, so this is probably a sequencing error. One nucleotide. And there's another one, and another one, and another one, and another one, and another one. So it's very likely that our sequence needs to be augmented with 16 residues. So basically, here, V, SV, all of that, this is missing in our original sequence. And that simply means that we have to transplant now this piece, which looks quite good, and to here. And there we have it. Yes. Final correction. Yes. So that V is in fact a V, not a G. And this is one of those cases where assembly of sequences, automatic assembly of sequences, results in a slight error. This may have been tolerated by the protein, I don't know. But I'd rather not find out experimentally where the protein is all bad and, crashy and stuff. This is not the first time and probably not the last time. So, exciting stuff. Based on the fold analysis, C-terminal histag looks appropriate. We're going to be expressing it in E. coli, so it doesn't seem necessary to put too much, additional stuff on the N-terminus. And also it doesn't seem to be necessary to put 8 his, 6 his should be sufficient. As you can see, the optimization was successful, not surprising, quantification x.9. We can just replace all of this fun with our sequence. Keep the stop codon. Keep 3 stop codons to be exact. And create a new translation, call it treZ, save, and here we go. 6 is treZ in a bacterial expression vector with a fixed up N-terminus. And we can save some degree of certainty. This was fixed up. Okay. All we have to do now is to design a series of cloning tools, a couple of primers here and there, to facilitate the cloning of our inserts that will be synthetic, probably ordered from twist. Nice and affordable DNA, usually good delivery times and so on. So certainly many companies do that, we just happen to like twist. IDT DNA is good too, but a lot more expensive. Anyway, let's design some primers. So we'll pick something similar to this, an arbitrary melting point of about 57 degrees. It's just annotated backwards. And yes, I could have used a primer, but in this case it's going to be 24 TAC RC. We need the reverse complement of that. Okay, that's our one. Excellent. And then the other primer, probably like 57 degrees or so. forward, also 57 degrees. We can optimize this temperature a bit more. Find 58. No. 24 tag F, forward, primer bind. See, so what will happen is we'll order a synthetic gene going from the end, including the primer, which by the way is 23, so it's enough, all the way to the beginning of the gene, also including the primer. This would be a synthetic gene that will then be what I used to call cold fusion, now it's called Neb quick assembly, whatever, will be cold fused into the vector that has been amplified with these primers, creating a long piece of DNA that will align. Then the nucleus will digest it from one end, digest it from the other end. The two single-stranded regions will then be lined up. Polymerase will fill any gaps, and then E. coli will ligate the bits. I don't think that NAB-mix contains ligase, although I might be wrong. In the past, it was a problem with patent conflict with the with the assembly Gibson method, and maybe now it's not. Either way, we're going to do it in E. coli. It will be super simple. The same exact process will be repeated for the other two genes. So we'll have a total of 5 synthetic constructs, 4 for mammalian, of which 2 will be secreted, and then 1 for E. coli cytoplasmic expression. Okay, so based on these properties calculator, which used to be at Northwestern and now it's at Bison, they sadly took the original page off. It's a really useful little calculator. The temperature, melting temperature of both of these primers is not actually 57. The salt adjustment, it's 67, which is fine. Modern polymerases are awesome like that. So these are the 2 primers and the treZ insert. Now we repeat this process for the other 2 genes. Okay, one last thing that I've checked is the presence or absence of putative glycosylation sites in the luciferase because it's being secreted. So far, nothing. And this is a fairly decent predictor, so I'm going to trust it for the time being. However, there's always that potential. So, in a nutshell, here's our treZ insert. Here's our decaf E. coli insert. Here's the human insert for decaf. And 2 F-look inserts in his and CS. Now I'm going to go order them and have a little time off for myself.